Hello students, in this session I'll discuss about models of speciation based on genetic drift and hybridization and polyploidy. In our previous session, we discussed about allopatric, parapatric, sympatric and stachypatric speciation. In the present session, I'll discuss speciation based on genetic drift. There are three theories under this. First, the genetic revolution theory, the founder flush speciation theory, and the genetic transilence theory. The genetic revolution theory was put forward by Ernst Mayr in 1954. According to this theory, a founder population formed from an ancestral population would have less genetic variability. In certain cases, the entire population could have been started by a single pair or by a single fertilized female. Natural selection acts on this founder population, and with time a new population is created which is diverse from the ancestral population, leading to speciation. The graph here represents Mayer's diagram of genetic revolution. The x-axis represents time and y-axis represents genetic variability. Consider an ancestral population A with high genetic variability. Due to genetic drift, basically founder's effect, the population goes through a bottleneck to form population B. Due to the bottleneck effect, the genetic variability of the population is reduced. With time, the population acclimatizes to the new environment and grows in size to form a relatively uniform population represented by population C. Increase in homozygosity occurs due to genetic revolution. As time passes, other evolutionary forces like mutation, recombination act on this population, increasing the genetic variability. Genetic revolution is driven by natural selection, and certain alleles get fixed in the population. With time, the population regains its variability with new genetic composition, population D and population E. In other words, genetic restructuring happens in population D and E when compared to its ancestral population A. However, in this model, Mayr was unable to explain as to how random drift led to speciation. Speciation as per founder flush theory. The founder flush speciation was proposed by Hampton Carson based on his works on Drosophila species in Hawaii Islands. The Hawaii Islands has about 800 species of Drosophila which are believed to have radiated from a single colonization. Certain species are endemic to the island. The youngest island is the Hawaii and the oldest is the Kau. The older islands are now submerged and found northwest, as shown in the figure. The arrows in the figure represent the number of founder events that have occurred with time. Example, from Cowl to Wow, 5 founder effect. From Mau complex to Hawaii, 15 founder effects. The numbers inside the island indicate the number of species found. The high endemicity of species in single island and occurrence of new species in geologically young islands provide evidence that speciation event is associated with colonization in newly formed islands. Carson felt that the marginal population could be more responsive to selection immediately after a founder effect, and the rate of evolution does not depend upon slow accumulation of newly arising mutations. The figure here shows the phylogenetic lineage of Drosophila species found in the Hawaii Islands, which could be correlated to the events of formation of new islands. According to founder flush speciation, a small population gets separated from a large ancestral population. It could be either due to founder effect, that is, colonizing a new environment, or repopulating the habitat after a catastrophe. Due to this genetic drift, the small population has less variability and different from the parent population. During this period, as the number of individuals in the population are less and competition is less, natural selection is relaxed. This leads to formation of new adaptive gene complexes by means of recombination. This period is known as the flush phase. The founder flush leads to genetic revolution and causes speciation. The main difference between Mayer's viewpoint and Carson's viewpoint is, according to Mayer's theory, after genetic drift, the genetic variability decreases and leads to formation of a relatively homozygous population, which later gets diverged due to accumulation of recombination and mutation. But according to Carson, after genetic drift, the population formed has genetic variability. This is because the ancestral population was a closed genetic system where recombinations were less due to co-adapted gene complexes. But after genetic drift has occurred, when selection pressure is relaxed, during the flush period, the recombination breaks these co-adapted gene complexes. Speciation by genetic transience. The genetic transience theory was proposed by Templeton in 1980, and it combines the theory of Mayer's genetic revolution theory and Carson's founder flush theory. According to this theory, after the founder flush effect, accumulation of inbreeding occurs which induces genetic disequilibrium, basically gametic disequilibrium. Selection occurs at multilocus systems relating to behavioral patterns, development, physiological traits, etc. If the founder population has sufficient genetic variability, rapid accumulation of inbreeding without reduction in gametic variability occurs. A few of the key genes, generally with epistatic modifiers which are responsible for reproductive isolations, change and confer reproductive isolation. This leads to a rapid shift to a new adaptive peak, and this is called as genetic transience. The table here lists the differences between the various theories of speciation based on genetic drift. Speciation by means of polyploidy and hybridization. Polyploidy is a rare phenomena in animals. It occurs often pathogenetically in organisms such as weevils, grasshoppers, salamanders, and lizards. However, polyploidy is common in plants. Speciation through polyploidy is the only mode of speciation which occurs instantaneously and the only mode of sympatric speciation. Second, hybridization. Sometimes hybrids formed between two species are fertile and are quite different or distinct from the parent species. In plants, many diploid hybrids, when fertilized, the progeny is reproductively isolated from the parent species and give rise to new species. As per Grant, this type of speciation is known as recombinational speciation. The best example for speciation by means of hybridization and polyploidy is seen in plants in case of evolution of the modern wheat Triticum asativum. Triticum asativum has three sets of genomes A, B and D that it has derived from the ancestral species. The A set of genome is derived from Triticum monococcum, the B set from the wild Triticum species. Fertilization of these two led to the formation of sterile hybrids AB. Doubling of this lead to the formation of the emmer wheat with genomic constitution AABB. Hybrids formed between the emmer wheat and the wild wheat Triticum taustucci with D genome led to the formation of a sterile hybrid ABD, which later by meiotic error and self-fertilization led to the formation of the present-day wheat Triticum asativum with three sets of genome AABBDD genotype. Thus, speciation results from various forces like mutation, recombination, natural selection, genetic drift, hybridization, polyploidy, geographical isolation, and reproductive isolation. With this, we finish the different models of speciation. Thank you.